Okay, it's 2:04, so why don't we start? I was going to end with this, but I'll start with it. And it's about the reporting on COVID. And an economics professor at Dartmouth and others looked at media coverage for negativity. The major media is 87% negative in their coverage about COVID. The scientific is 64%. International is 51% negative, and US regional newspapers are 53%. So the major media tends to portray things in a negative light. Not that there aren't negative things to report, mind you. And whatever we're talking about, changed some yesterday, could change tomorrow, and we'll know more months from now as well. But keep in mind that the media tends to present things either in a negative light, or if it's been negative for a while, then they've got to go positive so they can go negative again, because you've got to be reporting a change in order to attract viewers. So I just wanted to very quickly review viruses, just the basics again, so we know what we're dealing with. Viruses were only discovered in 1892, a tobacco mosaic virus, and it was a filterable particle that went through a very fine filter and still infected the plants. There's about 5,000 of them that are detailed, but it's thought that there are millions. They're independent particles with either genetic material of RNA or DNA, usually with a protein coat or some other kind of coat, maybe a fatty coat. And they have no energy producing capacity on their own. They depend on a host to reproduce. Some of them have outer spike proteins that help us to fight them because we can attach. They can be spread by respiratory means or gastrointestinal, like the norovirus, cruise ship, and the common diarrheal viruses, by blood with HIV, or even by insects like dengue fever. Their possible evolutionary benefit is that they transfer genes between species. But no one knows for sure whether they're always whether they're an evolutionary benefit or a detriment. People, by the way, are using the viruses now sometimes to introduce genes, and that's part of the vaccinations that with COVID. They're introducing genes to correct some defects, including some kinds of blindness or treating cancers. So this is a field that has really taken off in recent years, and there's a lot of hope for using viruses for therapeutic reasons in the future. There are 7 known coronaviruses, 4 common ones, and then the SARS outbreak about 15 years ago, the MERS outbreak about 8 years ago, and now COVID-19. How we respond to them is with either a quick burst of what's called the innate response. We've got factors that can be poured out that kind of non-specifically attack invaders, substances like interferon or cytokines, and they help neutralize it. That's one of the reasons why kids are less likely to get ill with this. Their innate response is very brisk. As you get older, we rely more on a targeted response with antibodies and other things I'll mention in a minute. But that's one of several reasons why kids do better. Their innate response is really very brisk. The more specific response that we get is from exposure to things that we get over time. And we develop either antibodies, which can glom on to the outside of the virus or bacteria or whatever other invader, an allergen, whatever. Those are produced by lymphocytes of a type called B cells. There's a couple different subtypes of that. And then there's a longer term memory that resides in a kind of B cell called a B memory cell, which was only actually discovered for sure about 20 years ago, but also the lymphocytes called T cells. And we'll come back to that with the vaccinations, because it's that T cell memory that helps you not get sick a year from now when you see coronavirus again, or 2 years from now, or 10 years from now. How many people remember having chickenpox a second time in their life? Nobody. We do get some isolated reoccurrence of the chickenpox virus if you get a case of shingles in a limited way, although that can be quite painful. But when you get an infection of something and you're sick enough with it for a period of days, you build up a lot of not only short-term antibodies, but also long-term T cell and B memory cells, so that you're often protected for decades and sometimes for your whole life. And that's what vaccines hope to duplicate. Now, you realize that the flu shots we get are every year. It's still hoped that there'll be a flu shot that will give you protection for many years. But one of the things that viruses can do is mutate, so that the flu shots has to be adjusted each year or every couple of years as things change. But also, unfortunately, and this is still puzzling to many people, the flu shots that we get don't give us a long-term immunity, not as much as was hoped, although they might carry over a little bit for a couple of years. So one thing about vaccinations also, just like the initial infection, and why you have to be careful with vaccinations is, and we'll get to this, the current topic, is that there's side effects, but not only that, there's sometimes that a first case of a disease or a vaccine may give you some protection. It turns out with dengue fever, which is a mosquito-borne virus, that your first case, you may get mildly ill, but the second case, your immune system is so strong, you really get sick with the second case. And that happened also with the dengue vaccine that came out, so they had to get rid of that. So in making vaccines, there's always the possibility that you might actually give somebody a heightened response to something that could be harmful. And we'll touch on that because of events also in the last couple of days. So one of the reasons why we probably get long-term protection is not just that we get a burst of a vaccine from the just for a day or a couple of weeks that stimulates it, but when you're sick, the virus runs through your whole body, you're sick for a number of days, you build up some long-lasting immunity to it, hopefully. And as it turns out, that the way that we build up long-term immunity is, if we're lucky, that the virus causes particles to appear on the to our immune cells in a way that they're able to attack it with antibodies or receptors that can be concentrated over a short enough distance that they can cross-link in a process that no one understands yet, but allows you to form long-lasting immunity. Now, the reason why that is important is that in theory, and often in practice, if you just give someone then say the spike protein, or in the flu shot that we get for adults each year, you're just giving someone the outer coat pieces, split pieces of the virus, so that it's not in a whole virus particle that allows our cells to attack it in a way that can cross-link and give us long-term immunity, as it turns out. So that's the second reason why the flu shots may have to be given each year. it's been hoped then that if you can give somebody as close to the natural infection, like giving them a whole virus particle, or at least a big enough part of the virus particle that our body can cross-link and form long-term immunity, that you'd get long-lasting benefits from vaccines. Some of the vaccines do that, like the polio vaccine and other ones. But the theory isn't always borne out in practice. And the measles vaccine, for example, which is very good and gives you immunity for many decades, actually was found to wane after 20 years or so. And even people that have had the measles vaccine can sometimes come down with it. There are occasional outbreaks. So that in part, the vaccines help, but it's partly herd immunity. So that if you vaccinate 70%, 90%, 95%, whatever it is, and you then don't have the virus circulating, you wouldn't be exposed to it, and then you wouldn't come down with a disease. But they came up with some outbreaks of measles in college campuses from people, often from international students, that brought measles in, and they found that actually their immunity had waned, and they just didn't know it because they were never exposed to it. So there's this trade-off all the time between the vaccines, hoping that they'll last a long time, and our body's ability to remember what it's seen, and hopefully the vaccines duplicating that. Now, the specific COVID vaccines are, you've heard a lot about. And in theory, the ones that will work the best long-term are ones that are whole virus. That is, you take a whole virus, like the FluMist, the spray that's given to people under age 50, that is a weakened virus, and it's the whole virus. So that you'd think, well, that would be pretty much able to duplicate what a natural infection would be. And you'd say, if that was the case, well, the FluMist vaccine would only have to be given to people every couple of years or 10 years or something, just accounting for occasional changes in the viruses. But that turns out not to be the case. And in preparing the talk, when I first gave it back in December, I researched several virology texts and kept searching online, and no one is discussing why the FluMist doesn't last for years. So there's a lot that is still not known about why vaccines work and whether they work better or worse. So there's the theory, and then there's the practice. So the whole cell vaccines then, in theory, might be better. Well, there's a number of different vaccines that are under study throughout the world. There's something like 70 or 100, and there's different mechanisms. One of them is a Chinese vaccine, the Sinovac, which is a weakened whole virus. They weaken these viruses by either treating them with some chemicals or using some kind of heat or other trick to denature them. And again, in theory, these should work to give you some long-lasting immunity, but these take longer to produce. For example, the flu vaccines that are passed through eggs and a series of steps, and eventually they identify a weakened virus. They're more cumbersome to produce. And also, since you think you've weakened them, but you don't know for sure until you've tested it in a lot of people, it's more dangerous in theory to produce these. So you have to be testing more people and watching out for some of them to revert to the actual active virus. And keep in mind that the oral polio virus does have that happen in about 1 out of 1,000,000 cases still. On the other hand, the weakened whole viruses like poliovirus give you some herd immunity, because the people that get it will actually be shedding a little bit into their surrounding environment, depending on whether they're coughing or sneezing or whatever, or not cleaning themselves well off the toilet. But you get some herd immunity from it. Whereas if you just give someone the inactivated virus with a shot, there's no herd immunity to it. So there's trade-offs always with these vaccinations. The exciting thing about the COVID vaccinations is that the technique for the messenger RNA vaccines that probably just about all of us have gotten was able to be produced very quickly, because they had been worked on for about 15 or 20 years and had some earlier vaccines against the SARS virus or the MERS virus, but those outbreaks petered out so quickly they didn't mass produce them and show that they worked on a large scale. They seemed to work on a small scale. So that's the Moderna and Pfizer vaccines. And they take the viral mRNA, that's the messenger RNA, that it codes for this spike protein and coats it with a fatty coat so that keeps it from getting broken down when it's injected into our systems. And otherwise, there are all these enzymes that we have that would break down these pieces very quickly. And that causes our bodies to make the spike protein and form antibodies against it. Now, we know in the short term, we form great levels of antibodies to this, and it gives us great protection. What we don't know is whether we're then producing enough of the spike protein on enough cells that are located close enough together, say, that our T cells can cross-link and form long-term memory from it. So we know that the antibodies from it, though, from the trials that were done back in last summer to the end of the year, the antibodies seem to last in good levels for at least 6 months. We'll see how they do after that. But the bigger question is, did they also give us T cell immunity? Now, to test for T cells is more difficult. To test for antibodies is very simple. You take some serum and you collect out the antibodies and you put it in some tube against whatever it is you're trying to get it to attack, the spike protein or whatever, and you can test to see whether that's happening and whether it neutralizes it. And these vaccines give us good levels of antibodies. The T cell testing to show whether things work is more complicated, and nobody can agree on the gold standard for what is an accurate T cell test. But a number of labs have been testing these vaccines, and each of the ones that come out, I'm sure, to see whether it gives you some T cell response. They put some T cells in a test tube, and they add some of the spike protein, and they look to see some reaction is occurring. And it seems to be that that's happening for a number of people anyway. And so there's great hope that we'll get some T cell long-lasting immunity from this, but we won't know that until 6 months, 1 year, 18 months, 2 years, 3 years go by as people continue to test it, because nobody has ever looked to see if these vaccines give you long-lasting immunity. It is said with the commonly occurring coronaviruses, which are some of the common cold viruses, that the immunity to it only lasts at most a year or maybe 2. That may not be true though, because I'm not sure that people have actually looked for T cell immunity. When we are attacked by something, we form antibodies, we end up having our antibodies be revved up sort of maximally about 10 days to 2 weeks or 6 weeks. And then they start to decline because it's a waste of time to keep producing antibodies for something that isn't bothering us anymore. So antibodies always decline after any infection that you've controlled and has gone away. It looks like the levels of antibodies so far to these mRNA vaccines, as I mentioned, are good for at least 6 months for most people. Now, other vaccines have other ways of trying to get us to react. Both the AstraZeneca and the Johnson & Johnson vaccine have an adenovirus, which is a common virus. They've each picked a virus vector of adenovirus that somehow should not cause us disease. In one case, they used a chimpanzee one. Doesn't cause people to get disease. In another, they used one adenovirus that was inactivated in a way, and AstraZeneca uses a second one for the second booster, which is also inactivated in a different way, but it's a slightly different virus, hoping they'll give us a better response. And both those vaccines have that virus carry in, instead of the messenger RNA, the viral DNA that causes us to produce, again, the spike protein. Now, the viral DNA means that those injections only start to produce when the cells in our body that have gotten that virus are actually doubling when they're multiplying. So that takes a little bit longer in order to be as effective. But both of those seem to be reasonably effective. The difference is that, though, each of those are using an adenovirus in order to carry the message into our cells. And as far as we can tell, but it's really early days, we'll come back to this, it's probably something to do with the adenovirus, which is triggering these rare reactions. So different vaccines causing you to produce the spike protein, but by different means. Both are pretty effective. We'll get into the effectiveness in a moment, but they are slightly different. The messenger RNA, the Pfizer and Moderna, again, the ones that are about 95% of the vaccines in the United States so far, were about 95% effective against getting symptomatic disease in the studies, and 100% about protecting against getting severely ill or dying, which is fantastic. I mean, 51% would have been decent. But those were in trials that were towards the end of last year. So since they've come out, though, the people that have gotten the vaccines have continued to be followed, and they look to be about 90% in real world effectiveness, and still about 100%, maybe not quite 100% against death, but pretty close to it, and about 98% against severe disease. So the other vaccines that came out had a high bar to compete against. Both the AstraZeneca and the Johnson & Johnson did not do quite as well, and that's caused a lot of people to be reluctant to take them to begin with. the AstraZeneca protected against moderate to severe disease by about 70%, against hospitalization, about 85% against death. It was about 100%, but there were only very few people that died in the placebo group, so that it wasn't quite as good. And it's not quite as good against some of the variants, one in particular from Brazil or South Africa, the AstraZeneca, for reasons that are not clear, didn't seem to do nearly as well as the messenger RNA viruses, although they're both asking you to produce this spike protein. So again, the theory would be that they'd both be equally efficacious, but it wasn't quite the same. Still, they were excellent, and the Johnson & Johnson had similar kinds of statistics. They still had excellent protection against severe disease or death, although you might get some symptoms from them. So there are other vaccines that are available around the world and going to become available also. As I mentioned, the Sinovac vaccine in China, which has already been distributed to a number of countries, that's actually an inactivated whole virus. The problem with the Chinese is they're not very forthcoming about their data. And they did publish one study that showed pretty good efficacy, but it hasn't been tested in such large numbers of people, and hasn't been as transparent. Similarly with the Russian, the Sputnik V, Sputnik 5 vaccine, that's also an adenovirus vector vaccine, which seemed to be pretty good, but the Russians are having problems with production, and as it turns out, they have about 7 or 8 different production facilities, and some of them are in other countries. And in Slovakia, they just got a shipment of vaccine that wasn't what the Russians had said that they were going to send them. I think they were milky, or they were single-use vials instead of multiple-use vials, and it turns out the productions from other countries may not be that reliable. Mentioning that, you realize that the Johnson & Johnson vaccine, they had a plant in Baltimore, which also was producing vaccines for AstraZeneca. And because this is all a big rush job in order to produce these vaccines, some of the workers in the factory were working on both vaccines, and they ended up with cross-contamination, and they had to throw out millions and millions of the Johnson & Johnson vaccine. So the vaccines themselves, though, you should be quite reassured, are better studied than any vaccines in the history of mankind. Now, even though they've been produced on fairly short order, the ones with the adenovirus vectors have been produced before, also for Ebola. Again, there weren't enough people to use in many thousands of people, but they seem to be very effective. They had the technology ready to go, and they're pretty effective. The other vaccines that will roll out have different mechanisms. There's one which is just the spike protein itself. Novavax is the company going to do it. And it's probably going to be effective in the short term, give you antibodies, but it's not so likely that it will give you long-term immunity. Again, in theory, and in practice, it doesn't always turn out that the vaccines that you're working on will do what you hoped. Now, keep in mind that Dr. Fauci and others have been working on a vaccine for HIV and also a vaccine long-lasting for a number of different flu types for over 40 years, and neither of them have succeeded. So that the proof to all this will be with the passage of time, because when I say it's been better studied than anything else, really in history, is that the number of vaccines that we're giving and the amount of information that we're getting afterwards is really unprecedented. Realize that the yearly flu shots are tested in about 6,000 to 8,000 people, tops. The reason is that they have a lot of experience with flu shots. They know the production, and they know that they've been proven to be safe, rare, bad side effects. And so they just test 6,000 to 8,000 people each year to see that they're getting adequate antibody levels in a number of different age groups. They say, okay, you're good to go. Well, with this, with vaccines now, many of you probably signed up as I did, that when you get your vaccine, you log into a system that asks you about your side effects, and it goes on every day for a week, and then every week for a couple of weeks. And so about 10% of people are signing up for that. So far, with about 100 million doses in the United States, there's about 10 million people that are giving feedback all the time about what's going on, which is totally unprecedented. And that's one of the ways that they've tumbled to that there are potentially some rare side effects from the Johnson & Johnson and AstraZeneca. They're doing something similar in Europe also. So as far as the usual side effects, we all know that you might get a fever, headache, something, or just feel generally tired for a day or two. A few people get a red arm about a week or two later for a few days, as their immune system probably is maximal at that point. But as far as anything more serious than that, there hadn't been anything until the AstraZeneca and the Johnson & Johnson, again, the two adenovirus vector vaccines, have been reporting some rare blood clot problems. Now, when I say rare, blood clots are fairly common. There's somewhere between 300 to 600,000 blood clots that are detected each year in the United States alone. In people that are taking oral contraceptives, 1 out of every 3,000 can expect to get blood clots in the course of taking them. In people that have COVID, sick enough to be in the hospital, 1 out of every 5 have serious blood clots. So the blood clots that the AstraZeneca and the Johnson & Johnson people are having, some of them have been just blood clots in the leg, which isn't so uncommon, but there have been some unusual blood clots that people have had. There have been blood clots in the brain in an unusual location in a vein there, and those can be very hard to detect, by the way. You have to be looking for it with a special technique, and I forgot exactly, but a CT scanner and MRI has to be set to look for that particular phase of study in order to detect it, because otherwise it just looks like a usual stroke. So they have clots in unusual places or in veins in the abdomen, which rarely people get clots in. Still, these are rare. In Europe, where the AstraZeneca vaccine rolled out, they've had 222 so far out of 34 million vaccinations. So that's one out of every 100,000 or so. But it's an unusual clot, that which is how it came to attention. with the Johnson & Johnson, just in the last couple of days, they've found 6 out of more than 6 million doses for about 1 out of a million. Now, nobody really knows what the exact incidence of these other rare clots are because they're so rare, nobody keeps track of them, but it seems like this could be real. As it turns out, almost all of them are in women, and in women under the age of 60, usually between about 18 and 49. And as it turns out, almost all of these are associated with a very low count of called platelets in our system. So that in addition to our red blood cells and our white blood cells, which includes a couple different types, including the lymphocytes we depend on for immunity, there's also platelets that are some of the factors, along with other clotting factors, that go to stop bleeding when we get a cut. Well, it looks like so far, but this is all preliminary, almost all of these cases, if not all, these people have antibodies that form against their platelets so that they clump their platelets and they end up clotting. If they clump too much in different areas of the body, they leave you with so few platelets left that are circulating that you bleed instead. So it's an unusual circumstance in which people are getting clots with very low platelets. This is similar to a condition which is known with, which is uncommon, but it occurs when people are giving the blood thinner heparin. And in those people, they rarely do form antibodies to their platelets also, and have a similar kind of problem. So although the incidence of these is very rare still, and the risk from disease is greater, it makes people uncomfortable to be giving it to people that are at low risk, younger people, and people that don't have coexisting diseases, where if they get COVID, they might get sick, but they're probably not going to die. But that's why people are pausing now with a Johnson & Johnson in order to look in more detail. But it's very likely that in the next couple of days, they'll say, go ahead, because the risk of the disease itself is still much greater, as it is for many vaccines. Realize that back in the 1960s or 70s, I think it was, that there was problems with the pertussis vaccine, and there were some kids that were getting rare side effects, seizures, high fevers, and people were saying we shouldn't be giving this vaccine. And yet the number of cases was still very, very low, and the risk of disease was 100, 1,000, 10,000, 100,000 times greater, whatever it was for some of the vaccines. And so it was decided that there be a vaccine compensation fund. So every vaccine has whatever it is, 50 cents or $1 or whatever it is that goes into this fund, so that if you get some rare side effect, and this is going to occur for these vaccines also, I believe, if you get some rare side effect, it's covered by a national fund, so that you can cover still the whole population to protect, realizing that there'll be a few rare people that get bad side effects of the vaccines. So all this is being hashed out, and all of it will be discussed, and how that will impact whether people are going to take these vaccines or not, we'll see. Obviously, some people are already reluctant to begin with, but it's analogous to vaccines that have been available for a while, which we've been using, knowing that there are some rare side effects still. To example, the oral polio vaccine, that still led people to use the vaccine. how this is all presented to the public, we'll see. But I wouldn't have any doubt that the vaccines are still going to be recommended. Perhaps like the Europeans are doing, they're limiting some of the adenovirus vaccines to people over 50 or over 55 or over 60, because their risk of disease is greater and their risks of the side effects seems to be less. Now, why is it greater in women and in people that are younger? Well, people that are younger have more active immune systems, and these are kind of autoimmune conditions. It seems like you're building up antibodies against something that ends up injuring you. Now, it turns out that women have always been the majority of people that have autoimmune diseases, and the young people and people that are younger are also tend to be more. And it turns out that women's immune system is more active than men for a couple of reasons, but we don't need to go into that. So it does seem like these side effects are almost all in women, and they're also under age 60. That's still being looked at, but is likely to be the case. And in passing, it may actually end up explaining why some people, by the way, who didn't get any vaccine, still have these conditions just spontaneously, and we never knew what caused it. But we may actually be finding that it may be something which is a side effect of some adenovirus infection that people had had, and we never tumbled to it until we gave millions and millions of people these kind of vaccines. But we'll see. Now, what about treatment? Suppose you do get the virus still. Are there good treatments? Not really, although there is some optimism. The benefits of treatment are still very low. Just approved in the last week was an antibody cocktail, which has been tested in people that had symptoms and were at high risk. They were obese, had diabetes, whatever, and they were already symptomatic, tested positive. They came in for one infusion of these antibodies, and they showed that instead of being in the hospital, say, eight people out of 100, it knocked it down to, say, 2 or 3 people out of 100. But most people still didn't get sick enough to be in the hospital. Well, just approved was to give people that antibody cocktail to people who aren't even symptomatic, but just live in the same household as somebody who's tested positive, because there's a big market for this. So they tested it in about 1,500 people, and they found that instead of instead of 3% of people ending up going into the hospital or dying, it was 2% of people. And this is approved now because the benefit's 1 out of 100. Well, it doesn't seem like that's much, but that's kind of on the order of what the treatments that we have do benefit people. If you're in the hospital already and you're sick and you get an antiviral medication, you'll get a medication called remdesivir, which decreases your time in the hospital by an average of 5 days, and not quite statistically decreases your chances of dying by 4 out of 100. If you're also sicker and you're in the intensive care unit and you get steroids, you'll decrease your chances of death. These are people that are really sick, from about 40% to about 30%. So it makes a difference in 1 out of 10 people. So it's not that there are no therapies that make a difference, but they only make a little difference. And by the way, that 40% death rate, that was earlier in the pandemic. Now the people that end up in the hospital, instead of it being 20 and 30% dying, it's more like 10% or so. And so what's the best thing to do still besides vaccination is exactly what they've been saying the whole time, and what Dr. Fauci still does himself. Don't travel to places where the virus is raging and variants are common and spreading. And that may be likely for the next 2 or 3 years at least, because the whole global population will have to get vaccinated or have herd immunity. Wear masks, use social distancing. And by the way, 6 feet is better than 3 feet, 12 feet is better than 6 feet, 18 feet is better than 12 feet. But it's not unreasonable to do 3 feet in public schools in younger kids, where they've shown that if they wear masks, the kids who said are less likely to get ill to begin with, so that these guidelines will continue to change. There's nothing magic about the difference, though, and that these guidelines will keep changing, and that herd immunity, we don't know for sure how much that will take, maybe 70%, maybe 75, maybe 80%. And realize there's somewhere around 30 to 40% of people at this point are not likely to get the vaccines in the United States. So that may be difficult to achieve. And what about how long is the immunity that we get from these pretty good vaccines that are already out there going to last? Well, we hope it's going to last for longer than 6 months. We know they're good for 6 months. How much longer? We don't know. So we will see. By the way, none of these guidelines and effectiveness also pertains to people that are immune suppressed, people that are getting chemotherapy, who have weak immune systems. They have been tested with the vaccines and they don't respond very well. Maybe 17% of them get decent antibodies. What about pregnant people? They weren't part of the trial, but there have been a lot of pregnant people that have gotten the vaccine and they've been pretty well tested. They get good levels of antibodies. The antibodies seem to cross into the placentas. They get some into the breast milk also. There's increasing evidence that it's a good thing to get vaccinated. Nobody can say that it's approved for that, but it makes good sense. Again, one thing is that these vaccines will continue to be extremely well studied. And the variants that come along, the Moderna and the Pfizer vaccines are still pretty good protection, probably good enough against those variants, which are increasing. The British variant, the B.1.1.7, is now the majority of new cases in New York City. The other variants are still rare in the United States. They are seen, but they're rare. We have a few cases of variants in this community. They're being tested at the lab at Cornell, but they're rare. And what about the long hauler patients? You'll see different estimates about how many are long haulers. One recent study that looked in England over time that showed 4% of people still had symptoms at 8 weeks and 2% after 12 weeks. But another study suggested that it was 50% of people that had symptoms that lasted for months. And of those, 80% had at least one symptom for 6 months. So it's not clear, but it is clear that there are some people that have lingering symptoms, fatigue, headache, kind of like chronic fatigue syndrome, which has been known actually for over 100 years, that some viral, bacterial, or protozoan infections cause people to have that kind of chronic fatigue, which has been recognized in the past 50 years for reasons that we still don't know, but seem to be in some people, their immune system still remains turned on and is making them sick is the most likely, rather than that the pathogen is continuing to spread. But again, that's all going to be studied. Surface contamination never made much sense. It's rarely a source, and the CDC finally agreed in the past week. Chances of it actually causing infection are probably about 1 in 10,000. You've got to blow a bunch of snot onto a surface, have somebody come by, swipe it up in their hand, put it in their mouth or nose pretty quickly, although it could rarely happen. It was never likely. Again, viruses don't live long outside the body. And unless you have a concentrated glob of snot that you're putting in your mouth or nose or something like some kid might do by mistake, you're unlikely to get enough of it that it would make a difference. Rare reinfections, people who got the vaccine or already gotten sick, can they get reinfected? The people with mild illnesses maybe don't have such a good antibody response. They can get reinfected sometimes, not often. Almost everybody else gets pretty good antibody levels. Reinfections are rare. They have been noticed. They've almost all been picked up incidentally because people are getting routine testing because of their job or travel, and they rarely are sick with it or at all, although there have been a rare case or two that have been sicker with the reinfection with a variant. That is all I think I wanted to touch on for now, but what questions?
I have a question, John.
Yep.
It's got to do with the AstraZeneca and J&J, both adenoviruses. So, okay, we hear a lot about what kind of clots we have with J&J. The US news has been great. I don't remember hearing very much about whether the AstraZeneca clots were similar types of clots with the low platelets. And have we been hearing that also about them? And then,
yeah.
then also
Yeah.
one corollary is, I sound like someone at the president's news conference, how would you treat this type of blood clot? But let's find out, is AstraZeneca having somewhat similar?
Yes, it seems to be a similar syndrome in which the platelets are clumping, and if you just check somebody's blood, their platelets are low.
Right.
So it's a similar mechanism. The way that they use to treat it, nobody knows. They're giving them something called immune globulin, which kind of swamps your immune system, helps turn off your own antibodies. And very cautiously, they might give them a different kind of blood thinner, not heparin, but they're worried about doing that because their platelets are real low because they could bleed. So nobody knows. They're collecting evidence on that.
Yeah, it's tricky. Okay, thank you.
John, if someone's been vaccinated and then hangs out with other people who've been vaccinated, we understand that risk is quite low. If you hang out with somebody who has not been vaccinated, is the risk of your becoming ill higher? Even though it might not be extreme illness, but still some risk. Is that a higher risk if the person is not vaccinated? And then there's the question of if you've been with someone like that, what are the chances that you can pass along COVID to someone else, even if you are asymptomatic?
Yes, that's a very important question. It has been partly studied, but it's not known for sure. That's why there is some hesitancy to say that people that have been fully vaccinated are not at risk to other people. It looks like when you take people that are doubly vaccinated and you just have them come in again, they're working, they're healthcare workers or whatever, and you swab them every week because they're at work. There are some people that do test positive again, but it's a very, very low percentage, something like 0.5% or something like that. And generally they have very low titers of virus, and they don't know how long they might be shedding virus for. So it's probably a little virus and not for very long and not very many people, but it's not exactly zero. And there are some cases of variants that you can pick up that you can maybe pass along a little bit more, so that the fully vaccinated people are at very, very low risk for at least 6 months of getting any disease themselves, and very low risk of spreading it to anybody else. But if you're around someone else, you've been vaccinated and they haven't, or anybody that's immune suppressed, somebody that's getting chemotherapy, you don't want to go over and visit a friend of yours, even if you've been doubly vaccinated, unless you've been super careful, you haven't been around anybody, and you wear a mask when you go there. You still have to be careful around anybody else that might be at risk of picking up asymptomatic spread from you. So there is some caution about that. The risk is very low. That's all we can say. But that brings up that you still need to be careful, even if you've been vaccinated. You can be, as the CDC says, you can be around other people that have been fully vaccinated without masks on. But you also want to be, I would say, the extra step to tell them that you should not now then be somebody that's working in a hospital that might be picking up, and then I'm going to go visit my mother who's getting chemotherapy. So you want to be thinking the second contact beyond you also, if you've been around anybody else that might have an asymptomatic spread.
Thank you.
I have one more question if someone else doesn't, and that is that I've been reading that the Chinese are admitting that their vaccine isn't very good. And even though we haven't seen a lot of their data, but they've been admitting that it's not good. Is there any I mean, they're the only one you mentioned who used the whole virus.
Right.
Is there enough knowledge about that as a methodology, or is maybe the way they made it, or do we not know anything because they don't give us any info?
We don't know. I mean, somebody knows somewhere. I tried to look up a package insert, some of those. I didn't find it for the, actually I did find it for the Sinovac vaccine, come to think of it, but I forgot where I've written it down. But as I remember, they inactivated it with a kind of chemical, and then they did something else to purify it. And there's different ways of inactivating it. And so, and nobody's ever looked that I know of into what happens to a whole virus when you inactivate it with heat or with cold or with serial passage or with a certain chemical treatment. Does that mean that it's still like a whole big particle, or is it broken up pieces which aren't so likely to give you long-lasting immunity, or might have changed the configuration of the spike protein somehow? They don't know. So it's disappointing that the whole virus vaccines don't seem to work as well as the natural infection for reasons that are not clear to me in looking at the textbooks that I've found. You have to ask some researcher for better clues.